So over the last 3 years, we've been taking soil samples from here at Hart. I've been taking them to Wait, where I've been processing them myself, and we've been trying to look at differences in the soil microbial population with a specific focus on microbes which are associated with nitrogen cycling. Hi, I'm Sarah Nowak and I'm the Research and Extension Manager for the Hart Field Site group, and we're here at Hart looking at two projects which Hart have funded by SAGET, and also Hart hosts a number of other SAGET funded trials with researchers from Adelaide who bring their field trials to Hart. So the specific projects that I've been talking about with the SAGET trustees today is our long-term seeding systems trial. So the trial itself was established in the year 2000, and it was very simply trying to quickly demonstrate and have a look at some of the influences that different seeding systems were having in the early 2000s, so the shift to one-pass seeding systems. And within the trial we have 3 treatments. One is a strategic treatment, which is a seeder which uses slightly wider tines, a no-till system which is pretty reflective of the seeding systems used in the mid-north, and we also have a disc seeder treatment. So for a number of years we've really been monitoring soil nitrogen levels. We've got 2 different nitrogen rates imposed over the seeding systems, but really we've been looking at maybe some different changes in soil nitrogen levels, how the crop growth has been going in terms of grain yield and quality. So a couple of years ago we realised that we were sitting on this asset of a really long-term trial, and it enabled us to perhaps look at changes in the soil that you wouldn't be able to measure in maybe a 1 or even a 5-season trial. So we put together a project which was specifically looking at soil nitrogen cycling with a specific focus on soil biology. So we were really looking at trying to unravel or get a better understanding of how the soil nitrogen might be cycling differently under the different seeding systems. How did that affect the microbial population?